Hey guys, welcome back. This is our third video and it is on cladistics, found in 2.6. Here's our term overview to give you a bit of an idea where we're going. A couple of key things to point out. We have our first assessment in week 8, that's the data test. Year 11s, you go on camp in week 9, and I will also be going on a camp in week 10 with the year 10s. Cladistics, an alternative system of classification. But first up, a disclaimer that we will be talking about evolution today. And evolution is not a new idea. It goes back to as far as 600 BCE from what we know, where there was a particular cultural group who were including evolutionary ideas into their religion. But Darwin came along a lot later and obviously made it quite popular through his research. It's also worth pointing out that there are two types of sciences. There's historical science and there's observational science. Today we're talking about historical science, and it's worth noting that historical science is very hard to prove right or wrong because it is in the past. Everything's gone. It's not observable, testable, or repeatable. So let's get into it. The purpose of today's lesson, we're going to talk about clades. We're going to look at a few assumptions associated with cladistics. We're going to look at molecular sequencing and how this is involved in cladistics. And then we will be practicing interpreting cladograms. So first up, to define the term clade. There's a few keywords that we're going to go through. So this is another type of classifying we've said already. We've talked about one type of classification based on this man's work, Carl Linnaeus, and the Linnaean system of classification works off physical features. Today we're going to look at the work of this man. His name is Willy Hennig, and in 1966 he came up with a system that classifies animals based on their evolutionary lines. So in this picture here, we can see the evolution of the human from the monkey with various stages in between. It's worth explaining how this comes about. Basically, there's shared characteristics. So the reason why all these organisms are together is because they share various characteristics. One might be that they have opposable thumbs. It's also worth pointing out that what makes a cladogram is that they all come from a common ancestor, and in this case, it's the lemur. As well as shared characteristics, you can see that different species branch off from a common ancestor. This is due to different characteristics between the species, which is also known as derived characteristics. It's a feature that sets members of the clade apart from other individuals. The clade is basically the group of organisms in question. Group of organisms believed to comprise a common ancestor. A cladogram is the entire graph that we are looking at. And it's also worth pointing out that these graphs are in relation to time. So humans are the most recently evolved. The rhesus monkey is the most primitive in the evolutionary line based on a cladogram. So there's a few assumptions associated with cladograms. A cladogram is really a hypothesis of how different species are related based on similar shared characteristics. When we're looking at a cladogram, you will see that those branches do come apart, but for this whole thing to work, there are assumptions we have to acknowledge. Number one is that all species on Earth share a common ancestor. that's a very big assumption. Number two, this process of bifurcation that takes place, and it's the assumption that different species have split off from that common ancestor at some point in time. And just to break that word down a little bit, bi means two, and to furcate means to split. So along the common ancestral line, it splits into two, where features vary. And thirdly, this process assumes that changes even occur in characteristics over time. So you may be thinking at this stage, that's a lot of assumptions to make, and in science, it's something we don't really like to do. We like to be able to observe and repeat and test these things. However, this is historical science we're talking about. In recent times, however, the process of cladistics, the system, has been refined as technology has developed. And there is this particular type of cladistics that has come about since the development of genetic sequencing technology. And it is called molecular phylogeny. There's a few key terms here that we'll talk about as we go. So you may have heard that humans have 46 chromosomes, or 23 pairs. Different species have different amounts of chromosomes, such as the chicken has 39 pairs, and the fruit fly has 4 pairs. The process of looking at chromosomes and assorting them into pairs is known as karyotyping, and the outcome is a karyotype. A karyotype is the number and appearance of chromosomes in each cell. So humans, like we said, have 23 pairs, with 1 pair being the sex chromosomes. So exam practice. Here is question number 1. A clade is a group of organisms, A, B, C, or D. This is from the final exam, the mock version of it anyway. You can pause the video here and answer this question. Cool, should have had time by now. We'll go through this in class. The last learning goal was to interpret cladograms to infer the evolutionary relatedness between groups of organisms. Before we can interpret cladograms, I have to show you that there are 2 main types of cladograms that scientists use. Note that the ones that you've seen already are simplified versions. They will look differently, and we'll see those in a sec. So the 2 main types, the first one is time-rooted, and it will look something like this. We have various species along the side here, and we have a common ancestor, and then that common ancestor will split into 2. We have branches, and we have branching points at which it splits. Another word for a branch point is a node or nodes. These are very important as far as being able to interpret cladograms. So let me go back here. To know if it is a time-rooted cladogram, you can tell if the species are all lined up in a straight line. For example, a question might ask you, infer whether species E is more closely related to species D or species A. The way in which we figure this out using a time-rooted cladogram is by counting the number of nodes. The way in which we do that is by tracing back the lines. So I'll show you how. Now, A node is again where it branches, not where it turns. So if we are looking at species E compared to species D, let's travel back through the lines and count the nodes as we go between them. So species E, starting here, follow the line, here we are. We've reached our first branch point, our first node. So that's number 1. Where's my cursor? Number 2, number 3, and there we are. So there were 3 nodes between species E and D. That was 1, 2, 3. Remember that. Let's compare species E to species A now. So if we go back through the lines, 1, 2, 3. In this particular question, both species E and D and species E and A had 3 nodes between them. The more nodes there are, the more distantly they are related. The less nodes they are, the more closely they are related. So in this particular case, they have equally 3 nodes, so they are equally as closely related to species E. Let's look at the second type of cladogram. It's a genetic distance-rooted cladogram. And you will notice an obvious difference here. Instead of all the species names being lined up, they are not lined up. And the way in which you can infer how closely a species are related in this type of gram is by just looking at the literal distance of the line. Let me give you an example. If this is the common ancestor between all these species, so from this one common ancestor, all these ones are said to have come from it. I could ask a question like, which out of all these species is most closely related to the common ancestor? We do not have to count nodes in a genetic distance-rooted cladogram. We simply look at the distance of the line. The longer the line is, the more distantly the species are related. The shorter the line is, the more closely those species are related. So if this was the common ancestor, the closest species to this ancestor would be equally these 2 in length, because we're just looking at a horizontal plane here. If there were a line going here, straight up, a line here, straight up, line here, straight up, line here, these species are furthest away from the common ancestor. So they're most distantly related, but these 2 species are the closest. So they would be said to be the most related, most closely related. With that in mind, let's have a look at a question again from the mock exam. There is a tree here, and it's asking us to, firstly, it's on large cats, and there will be a question that will come up in a sec. But the first thing that we should be able to do by now is recognize, is this a time-rooted cladogram or a distance-rooted cladogram? Well, the names are all lined up, so it's a time-rooted. To figure out how closely species are related, we're going to count the nodes. Remember, the more nodes, the more distantly related those species are. So the question is, identify which species, N. nebulosa or P. pardus, is more closely related to P. tigris. Justify your answer. So I'm going to highlight these for you, and I would recommend you to do this in the test conditions as well. N. nebulosa, it's actually a clouded leopard. P. pardus is an African leopard, and P. tigris is a tiger. Your job is to use this method to figure out which out of these 2 species is more closely related to P. tigris. You can pause the video here, and we'll go through this in class. Another question with another graph. So the first thing to do is to look at what type of cladogram is this. The names are not lined up, so therefore it's a genetic distance rooted cladogram. You're going to look at the literal length of the lines. Remember, the closer the distance, the more closely they are related. This is the same graph, but here with the questions. So, number 1, or A, what is one of the assumptions of cladistics? Infer which species is genetically closest to the common ancestor for the finches. These are all finches here. Interpret the cladogram to infer the degree of DNA similarity of all the species. So that question is asking you to refer to the values you were given here. 0 to 0.2. Remember, the further away, the bigger the number, the less they are related. And lastly, to identify 2 other types of evidence that could be used to support this. Again, pause the video here and we'll go through it in class. So that is all for today. With this video, practicing the questions and with your readings and the discussions we have in class, I think you guys will be all over this. All the best, and I will see you soon.